and thanks very much for the invitation to talk. This work's co-authored by Kevin Moore and Kristy Hobson from New South Wales DPI, and from McKay, who's at SARDI in Adelaide. What I'll be going over is a new PreDicta B assay for Phytophthora medicaginous, and that's the pathogen that causes Phytophthora root rot on chickpeas. We've just got our first year of results, so this is very much an update paper where we've got some initial findings and things are looking good, and we just want to share that. But we've got a couple of years to go before this test will be commercially available. We hope it will be commercially available. So I'm going to present some results to support these take-home messages that we think that knowing the soil inoculum concentrations of this pathogen will help with management, in particular to identify low disease risk sites, low disease risk paddocks, and secondly that from our work at looking at the detection capability of the method, it seems to be pretty robust and seems to be detecting Phytophthora in samples from farmers' fields. So a couple of reminder photographs about Phytophthora root rot. It often starts in low areas of the field that during a rain event get a little bit of waterlogging. And this photograph is from the 2015 season, which was a pretty dry season. So even in a dry season in low-lying areas, if there's inoculum present, you can get Phytophthora root rot. And the extent of damage in some seasons can be very large. So this whole area of the field here is lost to Phytophthora root rot for this variety here, Boundary, which is quite a susceptible variety to Phytophthora root rot. So this can be a very serious disease. Its hosts are chickpea, lucerne and sulla, and there's also some evidence that burr medics and some other medic species and a native weed called Rynchosia may also be hosts. We're going to do some glasshouse work on those hosts to try and firm up that information. But if you see burr medics in a field that's maybe going into chickpeas, there's our evidence to date suggests that there could be some risk of Phytophthora associated with those medics. And just to remind you from that first photograph, Phytophthora needs a high soil moisture content soil. So a saturating rain event with a little bit of pooling is what it needs to kick the pathogen off and go through its life cycle. There's a bit of a generalised life cycle here for Phytophthora. I'm going to be talking about oospores, which are a thick-walled resting structure. So that's when the host is, when chickpea and lucerne are not in a field, that's the resting structures that are sitting in the soil and they can sit there for years. When a susceptible host is present, they germinate, and in the long run they can either produce mycelium or zoospores, and that is what infects the root of the chickpea or the lucerne. Now, why I'm talking about this is because PreDicta B have developed this new assay for Phytophthora medicaginous, and in the past most of the assays have been designed around cereals. So I'm sure you're familiar with the crown rot work and root lesion nematode work. And in this case, the test is branching out to start looking at other crops in the rotation, so it's really good that chickpeas have been included in this and Phytophthora is on the list. So in the case of those other diseases that I mentioned, the information can be used to identify low-risk fields and maybe medium-risk fields where you choose a particular variety which is going to suit the disease risk of that field, or you choose a completely different crop because it's looking too risky. So in the context of Phytophthora medicaginous, we wanted to know whether the information about the inoculum concentration, which is related to how much DNA is in the soil, and that can relate to how much inoculum that DNA represents, could be used to identify disease risk and yield loss. So we had an inoculum concentration trial at Tamworth. It was sown in early July, and we used a Phytophthora root rot susceptible variety called Sonali, and we had 5 inoculum concentrations ranging from 5 right through to 4,000 oospores per plant. It was pretty dry in July, but in mid-August we got some good saturating rains, about 39 millimetres over 3 days. Following that we got some hot dry winds and we got some rapid disease expression. So you can see here, here's our nil rate here. Still quite green, and we've got losing colour here in these higher rate plots. And I'll just move on to some September disease incidence results. So quite large differences. Here we've got our oospore rate from 0 through to 4,000 and our percentage of diseased plants at the end of September after these hot dry winds. How did they separate out? We had statistical separation between the 0 rate, which had about 10% infection, the 100 oospores per plant, and among the 500 to 4,000 oospore plants, there's no statistical difference between that group, so they're all pretty much the same and ranged around from 90 to 100%, so very severe infection in that high inoculum treatment. So moving on to October, a pretty dramatic photograph here of how things looked at later in the season, and I didn't think I needed to put a red circle around these zero inoculum treatments among our 6 replicates because they just stand out as those green plants there. So you've probably got some idea about what this means for yield. I'll just quickly show you the yield results. Our 0 inoculum plots had 1,248 kilograms per hectare. Our 100 had 38 kilograms per hectare, and these fellows here ranged from 4 to 0.5 kilograms per hectare, so very severe yield losses. So that was a trial with Sonali, as I mentioned, a susceptible cultivar, and it was interesting and of relevance to note that the higher inoculum concentrations didn't have any differences in the amount of disease and didn't have any differences in the amount of yield. That may be because during that period of 3 wet days that the low inoculum concentrations, the pathogen can reproduce, and so the lower, the 500 and 1,000 rates actually catch up with the highest rate, or it's just a matter that the amount of available root material gets saturated equally as well by 500 oospores as it does by 4,000. So there's no clear separation at those higher inoculum concentrations, and that could be due to another number of factors. So moving on to the implications for how this test may be used, these initial results suggest that the test has good potential for identifying low-risk fields that have no Phytophthora medicaginous inoculum. So that's useful, but it would be also of more use if we could also perhaps look at how we could use some fields that have a little bit of inoculum present, and that's where we want to consider varieties such as Yorker, which has improved resistance to Phytophthora medicaginous. So in our future work, we're going to start looking at varieties with improved resistance, and we hope that low Phytophthora paddocks may also produce low disease risk and good yields with these varieties. So that's where we hope that the use of the test may be applicable. I'm going to move on to the second part of our work now, which is looking at the detection capability of the test and how reliable it was at detecting Phytophthora medicaginous. We want to compare the PreDicta B DNA results versus baiting. And by baiting, I mean that we're growing a susceptible variety. So we have the roots of the susceptible variety within the soil sample, and we're wanting to basically catch the pathogen and isolate out the pathogen and identify it under the microscope so we know we've got Phytophthora medicaginous, and compare that result to our baiting result. So here we've got Sonali seedlings growing in soil where that was flooded. You can see the flood line there, and we've got some cankers forming, and we isolated Phytophthora medicaginous from those cankers and plated them out and identified them. We did this with soil samples that came from northern New South Wales and Queensland that were collected in 2013. And just a couple of points that affected our results. I need to tell you that those samples were stored in a chiller for a year at 5 degrees, and that most of the samples were collected by taking 80 PreDicta B cores per field. But for 5 or 6 fields, there was drought conditions and we couldn't get the core into the ground, so some large clods of soil were collected with a trowel. And in that case, the sample might be 5, 6 or 8 kilograms, quite large samples. That affected something that we found later on. That's why I'm mentioning that. Of those samples, we sent 400 grams off to SARDI in Adelaide for the PreDicta B test. So how did these results work out? We had 48 fields. The DNA results, the PreDicta B results, 11 of those 48 fields came back as positive for pmDNA, 37 were negative. Our baiting results, 10 of 48 gave isolates and 38 did not. How did these break down? Well, of the 11 that were positive for DNA, 9 of these gave us isolates and 2 did not. This might be interpreted as a false positive. And for the negatives, we had 1 of the 37 that gave us an isolate, and this might be interpreted as a false positive. The negatives are 36 out of 37. So I'm just going to dwell briefly on this result here, which would be of a concern to a farmer who has a result that says there's no Phytophthora medicaginus present, but then the disease turns up. That sample turned out to be one of our 6 trial collected samples, which were about 6 kilograms of soil. And what we think that, even though we tried to homogenise the soil very carefully, we think that when we took out the subsample for the sending to SARDI and we took out another subsample for using in the baiting, that we didn't have the same, we ended up with different results due to this subsampling error. And it's factors like this that can affect our results. And here we may have had some DNA present, but in quite low concentrations, and through storing the soil for a year, that could have affected these results as to why we didn't get an infection from these 2 samples. So as I mentioned, we think we've got a few subsampling and storage effects, but if you look at the PreDicta B guidelines, what they say is that you collect a sample and you send it off within a couple of days of collection. You don't store them for a long time, and another recommendation is not to subsample. So we did these things for special reasons because we needed to, but if you're having a commercial sample, you'd be sending it off within a couple of days, you wouldn't be storing it, and you wouldn't be subsampling it like we did. So we don't think that you would encounter these problems with commercial samples. But we're going to be doing this again this year with more samples, and we'll be taking a good look at that. In conclusion, we had a fairly good news. We had an 82% and 97% agreement for our positive and negative results respectively. So that's pretty good for our first year results, I think. So finally, our take-home messages. We think that knowing the inoculum concentrations of Phytophthora medicaginus is useful, particularly for identifying low-risk sites that then have low disease and a reliable yield. And we think, secondly, we think that the test is quite good at identifying Phytophthora medicaginus in soil samples from growers' fields. We've got a couple of issues to sort out, but the preliminary results look good. Thank you.